Good afternoon. Good afternoon. This session is about the immunodeficiency disorders or IDs. We all are aware of the factor that the immune system helps our body to protect against infections and the invading pathogens. So, if anything, any alteration or any change happens to our immune system that results in immunodeficiency disorders, that is, immunodeficiency disorders are associated with the defect or impairment in immune function. That means if our immune system fails to respond to the foreign antigens or foreign substances or foreign infections that cause immunodeficiency disorders. So as a result of that, our body will be susceptible to lot many infections. It is induced through infections or various environmental factors. Actually, the why immunodeficiency disorders are happening is it's not that very clear because some environmental factors cause immunodeficiency disorders. Few genetic factors are there, but not merely it is genetic. It is few genetic factors are there, then environmental factors also contribute to the immunodeficiency disorders. So, the immune system has to work together to defend against the infectious agents. This is called the immunodeficiency disorders can be classified into two categories, that is primary and secondary immunodeficiency disorders. The primary immunodeficiency disorders are caused by the intrinsic defects in the cells of immunity. That is, primary immunodeficiency is congenital, that means by birth, resulting from the genetic defect. So primary immunodeficiency diseases are caused because of the genetic factors, or you get those diseases by birth. That is primary immunodeficiency disorders. Then the next is that is it is inherited. Next type is secondary immunodeficiency disorders, or it is acquired through the lifetime of the individual. So in the during the lifespan he acquires it. By birth he won't get it, but that is what I said. That may be due to the environmental factors, or it may be due to some infections which happens to your body. So secondary immunodeficiency disorders are developed as a result of the abnormalities which are acquired through the lifetime of the organism, of the individual. Examples are HIV infection, maybe because of malnutrition, or maybe because of the immunosuppression, or it may be caused due to the side effects of some drugs or some that is the what is the tablets or anything taken. That is secondary immunodeficiency diseases. So immunodeficiency diseases are of two types, primary and secondary. Primary is congenital, means by birth. Secondary is acquired during the lifetime of the individual. So the primary immunodeficiency disorders, or this is because of the is caused when the four components of the immune system are affected. That is, they occur when there is defect in any of the four components. Which are the four components? They are the T cells. Primary immunodeficiency diseases is caused because of the deficiency or disorder in the T cells, maybe because of the B cells, phagocytes, or the complement system. So it is caused by defect and because by birth also itself that individual will be having defective. You can see here that is defective B cell or defective T cells, and as a result the complement system will be not be working properly. So that is the primary immunodeficiency disorders. I'm sorry, the fact or the components which are affected are the T cells, B cells, then the phagocytes and the complement system. And it is as a result of this primary immunodeficiency disease. What happens is by birth itself, that child will be deficient, or he will not be having certain the cells which work in immunity. As you all know, the cells which are working together in the body are the B cells, the T cells, and the complement system. In addition, we have many phagocytic cells which kill the foreign invading antigens. So by birth itself, if a child is deficient or with having defective cells, that child will be susceptible to many infections, and that may lead to the reduced growth or stunted growth of the child, or at times that child will lead to that these diseases will lead to death as a result of this defect in the or a reduced number of immune cells. That child will be susceptible to diseases, and that will lead to the death of the individual. And let's see the manifestations. That is what I said. The disorders are actually B cells, T cells, phagocytic cells, and the complement system. And some of the prominent manifestations and manifestations or the symptoms by which you can identify are some of the cutaneous infections. Some child will be having, and I'm not telling all the rashes on the skin, some recurring rashes on the skin, or some rashes or some pigmentation on the skin will remain as such for long. So that will not be some rashes may come because of some itching or something like that, but the rashes which is associated with the immunological disorders will persist for long time. So the manifestations of the immunological disorders are the dermatological conditions such as eczema and cutaneous infection, maybe like rashes or some black marks on the skin which persist for long. That is actually the first manifestation, one of the manifestations. And the symptoms which is associated with the primary immunodeficiency disorders are recurrent respiratory infections. Why? Because that is what I said, because that child will not be having some immunological cells, that is B cells or T cells will not function properly, and that as a result of that, the child will be susceptible to all the infections which is around him. And the symptoms are recurrent respiratory infections, persistent bacterial infections, sinusitis, bronchitis. Then every time the child will be having fever, cold, like that, then skin and membrane functions, resistant oral ulcers, conjunctivitis, diarrhea. As a result of this, small agents, small microbes in the food materials and water will lead to diarrhea and malabsorption. Continuous diarrhea will lead to the stunted growth of the baby or that child, because he is not having any immune cells to fight against the foreign antibodies. Some of this disorder of that is the primary immunodeficiency disorders are, one typical example is the X-linked Agammaglobulinaemia or XLA or Bruton's disease. And this is X-linked, that means it is congenital. By birth itself, the child acquires it. Then the deficiency of B cell, and this X-linked, the XLA or Bruton's disease is caused due to the deficiency of the B cell tyrosine kinase enzyme. And because of actually the enzyme which is lacking in the baby's or the child's body is tyrosine kinase. Tyrosine kinase will be lacking in his body. Why tyrosine kinase is lacking? Because the gene which is responsible for the production of the RNA, that is responsible for the production of protein, tyrosine kinase, will be malfunctioned. See, the gene which is responsible for producing this protein. Which protein is it? Tyrosine kinase. So tyrosine kinase will be lacking. What happens when tyrosine kinase is lacking? That result in the failure of development of B cell, or B cells will not be properly matured, or defective B cells will be formed. That is X-linked. X-linked means it is associated with the X chromosome. So from the name itself, you can understand it is congenital, by birth it gets. Which protein is not synthesized in his body? Tyrosine kinase. So the gene which is responsible for the production of tyrosine kinase will be malfunctioned. As a result of that, the maturation of B cells will not take place properly, and that will result in the disease condition called Agammaglobulinaemia or XLA or Bruton's disease. And in this case, the that is the males, the affected males have few or no B cells in the blood or lymphoid tissues. The male babies will not be having the proper or functional B cells in his body or lymphoid tissue, and consequently what happens is that the lymph nodes become very small and the tonsils will be absent in these individuals, and their serum usually contains no IgA. IgA will be lacking. IgA is lacking in case of these individuals. And for the first 6 to 12 months of life, these people may survive properly. So, always the congenital disorders can be, usually, the congenital disorders can be noted only after 1 year of the child. Why? Because for 1 year, he will be getting this immune cells. So, for the first 1 year of life, they will be protected by the passive immunity which they get from their mother. So, after 1 year only, we will be able to diagnose or the whether the child is having any disorders. Usually, after 1 year, because after that only his body start producing this immune response cells. So, what happens as a result of gamma aminoglobulinemia? That child will not have functional B cells, and this will be manifested only after 1 year. Why? Because that child will be getting passive immunity from the mother, and that will protect the baby for 1 year. And as after that, it will be vanished and he will be susceptible to recurrent infections. The patients with IgA deficiency will have very less amount of IgA levels. Next is selective IgA deficiency disorder. And this selective IgA deficiency disorder is one of the most common immunodeficiency, and it affects about 1 in 700, and usually that is found in the Caucasoid race or the white skinned people. And people with this disorder have very low amount of IgA. That is why it is called IgA. IgA is, that is immunoglobulin A, is deficient in this people, and they have very less because of the deficiency of this IgA. These people will be susceptible to diseases like chronic otitis, sinusitis, and usually they will be susceptible to this respiratory diseases. So, those children who are having immunological disorders will be continuously having some coughing, sneezing, and all. Continuously, if it is there in a person, he has to go for approach a medical expert. And people with IgA deficiency will develop type 3 hypersensitivity. We have learned about type 1, 2, 3, and 4 hypersensitive and sensitivity, and in the previous session. So IgA will lead to type 3 hypersensitivity, and their manifestation is they have respiratory disorders as well as they have diarrhea. Now, they are susceptible to conjunctivitis, then various gastrointestinal disorders, etc. Next is disorders of cellular immunity. Disorders of cellular immunity are T cell deficiency, that is, the T cell deficiency, and we have already discussed about the B cells, next about the T cells. The T cell deficiencies result in severely impaired cellular and humoral immunity, and these patients will have a recurrent life-threatening viral, fungal, mycobacterial, and protozoan infections. And one of the common examples are D. George syndrome, then combined immunodeficiency disorders. So, this combined immunodeficiency disorder, from the name itself, it is clear that combination, so deficiency of some of the lymphocytes will lead to combined immunodeficiency disorder. Hope this is important. Severe combined immunodeficiency disease or SCID, SCID. Combined immunodeficiency means combination of immune cells are non-functional. That is why it is called combined immunodeficiency disease. A combination of immune cells such as B cells or and T cells or IgA, IgG are deficient. Not only B or T but the combination. That means B plus T or IgA plus IgG and IgG. Few of the immune cells will be deficient. That is why it is called combined immunodeficiency disorder. So, combined immunodeficiency disorders result as a result of a combination of the humoral and cell-mediated immunity. Combined immunodeficiency disorder is caused as a result of, so combined immunodeficiency disease is equal to that is equal to it's caused as a combination of humoral and plus cell mediated. Humoral and cell mediated. That is B cells and T cells. Humoral and cell mediated cells together will be lacking. That is why it's called SCID or severe combined immunodeficiencies. And as a result, that is, and it is associated with, and the process of lymphocyte maturation usually takes place in the second slide I have shown. Usually maturation takes place in the stem cells, and the proteins which is required for the maturation of these lymphocytes will be absent in these individuals. Severe combined immunodeficiency means by birth itself they will be having defective genes that are responsible for the production, or they will be will not have functional genes which is responsible for the production of proteins that help in the maturation of this lymphocytes. Then the combined immunodeficiency disease or SCID, it is associated with that is children failure to thrive, and they will be having chronic respiratory disease. And maybe in your another chapter you might have learned in detail about skin. There is a child called De Silva, Ashanti De Silva, who was having a SCID, and one of the treatment is gene therapy is itself. And the other part of the immunological chapters you might have learned about Ashanti De Silva, a child who was having SCID. And that child cannot go to school. Why? Because when she goes to school, she has to wear mask, she has to continuously sanitize her hands, etc., etc. Even though small children, if they go out of the house, they will not follow such strict instructions or all. So she will be susceptible to various infections like viral, bacterial, fungal or respiratory infections. And so it's very difficult for those people to thrive. And they will be having persistent or recurrent diarrhea, respiratory infections. And SCID is actually managed to some extent. It is managed or it is treated by giving IgG infusion, or the stem cell transplantation and gene replacement is the best therapy for these people. That is, the gene therapy is done in the case of the child which I told, that is Ashanti De Silva, and she has survived after that. But the diagnosis is very important. After 1 year of age only you will be able to diagnose. That is severe combined immunodeficiency disorder, means a combination of immune cells such as B cells as well as T cells will not function properly. Now, next is acquired immunodeficiency syndrome or AIDS. acquired immunodeficiency disorder or syndrome or AIDS. Now, HIV stands for human immunodeficiency virus, or it's caused by a virus which affects the immune system of humans. And AIDS stands for, and usually for your exams you will be having a question to expand AIDS or expand HIV. So AIDS stands for acquired immunodeficiency syndrome. So what is a syndrome? Usually you might have learned about Down syndrome. You have learned about Down syndrome, Turner syndrome. Can you? I don't know whether you will be able to put the answers in the chat box. That syndrome means, because in we call Down syndrome, we call Turner syndrome. What is it? Because for a child who is having Down syndrome will be having lot many disorders. He'll be having some abnormalities on the face, genital disorders, etc. So, what a syndrome is? Syndrome is actually a group of symptoms. That is what is known as syndrome. So let's see about detail about the in a descriptive attitude about descriptive what HIV and or AIDS is. And AIDS is caused by a virus, HIV virus, and this HIV virus is a retrovirus, and these viruses multiply inside your body cells and cause illness. Like common viruses like the COVID-19 or measles, hepatitis, common cold, SARS, etc. So that is what I said. A syndrome is a group of symptoms and signs. Syndrome means a group of symptoms and signs which is associated with the same underlying condition. But why it's called syndrome? Because not only, for example, if you are having what any infection or flu, or if you are having cold is only one symptom. Some diseases will have only one symptom, like cold. You will be having running nose, that is a single symptom. But some other disease will have what? That is the case with SARS. You have a lot many symptoms, like you have respiratory problem, you will be having body pain, then you will be having headache, you will be having joint pain. That is, if too many symptoms are there, if more than one symptoms are associated with a disease, that's called a syndrome. So syndrome is a group of symptoms and signs which is associated with the same underlying condition. We have learned in human genetics, that's what I said. Down syndrome, that people who are having Down syndrome does not have a single symptom. They have lot many symptoms. Then Turner syndrome, many symptoms are there. You learned about a list of symptoms. That is group of symptom is called a syndrome. Syndrome means a group of symptoms or signs which is associated with a disease. So, a variety of opportunistic infections and cancers cause a variety of symptoms and signs. Now, let's see which part of this is affected. So, the infection is spread mainly as by sexual contact, through blood, blood products, etc. And the cells which are affected are the CD4 cells or CD4 and T lymphocytes. So the virus is a RNA virus in the group of lentivirus. It is a retrovirus because it produces an enzyme called reverse transcriptase. That means a reverse transcriptase is an enzyme which can produce what? DNA from RNA. Reverse transcriptase or reverse transcription in the reverse manner. That is why it's called reverse transcription. What is transcription? DNA to RNA. What is reverse transcription? RNA to DNA. So, retroviruses can produce DNA from RNA. Then, there are two major forms of HIV. Then, the and the primary targets of this HIV are, then HIV is the CD4 cells. That is, both the humoral and cell-mediated immune responses occur in HIV infection persons. That is, the early response to HIV infection is in fact similar to the immune response of other virus. Whatever happens, this is also a virus, like common any virus. it will produce some immune response in the body to remove it from our body. Once this virus enters our body, our immune system will fight to kill it at the earliest. But it is very difficult. We cannot destroy this virus once it enters the body. It is very fatal condition and it will suppress the immune system in your body. And the immunopathological effects of the HIV infection is related to the interaction of the virus with the receptor, and the target cells of these viruses are the CD4 cells. The CD4 cells on the cell surface. CD4 cells on the cell surface are the targets of these HIV virus. The CD4 cells or the T helper cells. And as soon as this HIV enters the body, it binds with the dendritic cells, and this dendritic cells will carry the virus to T helper or CD4 cells. And this is the structure of this RNA virus, or this is the structure of a HIV virus. It is, you can see it have a small RNA genome. The genetic material in the body of this RNA, this is the RNA virus. HIV is a RNA virus. Why it is called RNA virus? Because the genetic material is RNA, not DNA. And this RNA codes for, this RNA have 9,300 base pairs. Look at the structure of this virus. It have almost a spherical shape, and usually, and you are very much aware about the structure of viruses nowadays because of this is also having a structure which is similar to that of the coronavirus itself. It have large number of spike proteins on its surface. Then it has almost a spherical shape, and the viral genome encodes almost three major open reading frames, that is GAG, POL and ENV or envelope. GAG encodes a polyprotein, then POL encodes three important enzymes. Then there is another, the third major open reading frame is ENV or envelope, which codes for the production of this envelope of the virus. So see, look at the structure of this virus. It have an outer envelope, it have some spike proteins, it have a lipid membrane, it has a genetic material, that is RNA, and associated with the RNA there are proteins called protease. Then the main enzyme which is present is the reverse transcriptase enzyme, and the reverse transcriptase enzyme helps in reverse transcription. And another protein involved is integrase, which help in integrating the RNA, integrating this virus onto a host. That is integrase, protease enzymes. Sorry, proteins are there. The infection begins when this glycoprotein or the GP120 of the virus begins to bind. Here you can see this is GP120. The infection process begins when the glycoprotein GP120 of the virus binds the principal receptor on the target cell, that is CD4. The GP120 will bind on the CD4 molecule, the CD4 protein. Here you can see this is a receptor, the CD4 molecule. It binds onto the receptor, that is the CD4 on the cell surface, and this induces the conformational change or change in the structure within the GP120 protein. This binding helps in the altering or changing the structure of GP120 protein, and this altered, what happens? First step, GP120 binding on CD4. Second, what happens? There is a conformational change, means a structural change happens within the GP120, and this exposes the binding site for the co-receptor called the CCR5. See here, this is a co-receptor, and this exposes the binding site for the co-receptor or CCR5, and this results in the fusion of viral envelope with the target cell membrane. As a result, it will be integrated into the cell membrane. Then, once the viral envelope has fused with the target cell, as a result, this is it started. It's ready. So it entered the cell, the normal cell, and the viral envelope has fused with the target cell membrane now. See, fusion of this virus takes place, and the viral envelope fused with the target cells, and as a result, what happens? The contents of the virus enter the host cell. This is the mRNA. The mRNA reaches or enters the cell and the infection is achieved. And the reverse transcriptase enzyme copies the viral RNA into DNA. That is the second step. So, next step, the reverse transcriptase is a protein which is present in the RNA itself. So, the reverse transcriptase enzyme is associated with the RNA, and this reverse transcriptase enzyme copies the viral RNA into the DNA. Here you can see. So this copies, that is the reverse transcriptase enzyme is present, and this genomic RNA, with the help of this transcriptase enzyme, see here, it is integrated into the nucleus and it joins with the DNA of the host. So the integrated proviral DNA is result. So as here you can see the RNA. So the host will be having RNA plus DNA. Then next is the DNA copy of the viral genome is readily transported to the nucleus. See, from and it is integrated in the host cell genomic DNA, that with the help of the protein called integrase. Hope you remember in the picture I told there is a protein called integrase, and integrase help in integrating the RNA with the DNA. Then once integration is completed, the viral genomes can be transcribed by the host cell. So now what happens? It will multiply along with the DNA of the host. Then the next step is virion assembly, and it is followed by the processing. Now here you can see the genomic RNA and the protein synthesis, processing and assembly. Then the genomic RNA is now mature and it will come out of the cell for further infection. So first step, what happens? The glycoprotein or GP120 binds with CD4 on the cell surface. Second step, conformational change of CCR5, and that is CCR5, that a conformational change occurs. As a result of this conformational change in CCR, the RNA enters the cell, that is the genomic RNA with the proteins such as integrase, protease, etc. is there, and the RNA is there in the cytoplasm and it is entering into the host cell and it is integrated into the DNA with the help of integrase protein, and it will multiply, and this will multiply in the cell and it will bud or mature HIV virion. We have learned about virus, virion, etc. Our HIV virion is formed. So, during this course of infection, the T helper cells are lysed. Lysed means the T helper cells will be breaked. Lysis means the cell wall will break and new viruses are released. So, these viruses, here you can see the HIV, more viruses will infect more T helper cells, and this virus will multiply very fast, and the massive number of virus, that is more than 1 billion of virus will be formed one day, and this T helper cells, see, this much number of cells will affect all the T cells in the body. They will be running through the bloodstream and they can infect all the T helper cells in the body, and they will be, because it will take many years, because our body is producing T helper cells every now and then. So it will take some time or years for the virus to destroy the T helper cell. So the T helper cell population will be depleted or destroyed. First manifestation is the number of T helper cell. As a result of HIV infection, what happens first? the number of the T helper cells will decrease. See, the HIV virus multiply in your body. It will go on destroying the T helper cells. So it will take years. So it will take years for the virus to destroy the T helper cells. So the first manifestation is the number of T cells in the body will be decreasing. And the long-term asymptomatic period or non-symptomatic period of HIV is called a latent period. There's a time for every viral infection or every bacterial infection, there is a period called latent period. See, once the virus reaches your body, there will not be any symptom which is manifested outside. There is a silent time after the infection that is known as latent period. So, the latent period of the HIV infection will depend on the healthy condition of the individual. That's very important. If a HIV infection happens to a very healthy individual, no manifestation or no symptoms will be shown by that organisms, that individual. It will take some time. So it may take from 5 to 10 years or 2 to 10 years, or in unhealthy individuals it will be manifested within 2-3 years or so. And that silent period or non-symptomatic period is called HIV. It's called the latent period. And the clear symptom will appear only towards the end of this period. So when the symptoms start appearing, what happens? Almost all of the T cells might have be destroyed. So what are the symptoms? Because I said which cells are affecting? T cells. T cells number will come down. So when the T cell number come down, what happens? That person will be susceptible to infections. HIV, the symptoms are that individual will be, whenever the immune system is weakened, what happens? That individual will be susceptible to lot many infections like cough, cold, diarrhea, or food poisoning, anything, any virus or bacteria come on his way, it will catch up that individual, and and the people will be that person will be very weak. Then the T cell function will be non-functional. So the modes of transmission are through sexual contacts, then through the contaminated needles and syringes, the contaminated instruments, blood and blood products, then from mother to child transmission during birth. And these are the modes of transmission of HIV. Now, next is autoimmunity. Next topic is autoimmunity. Autoimmunity means our body will fight against ourselves. That is what is known as autoimmunity. It is a very dangerous situation. Autoimmunity is actually an abnormal response of our immune system. Auto means self. Our body itself will fight against our body. Our immune cells will fight against our cells. That is known as autoimmunity. It is a very, very dangerous condition. Then, autoimmunity is the abnormal response of the immune system against the self components, or it is a failure of an organism to identify or discriminate between the self and non-self proteins. The best thing our body can do is, how do we respond against a foreign pathogen? We say that it's not ours. But if our body cannot distinguish between the foreign and the self proteins, that is very dangerous situation. It will damage our cells itself in the body. And the basic feature of this autoimmunity is our cells will be damaged by our cells itself. The immune system normally responds only to foreign antigens, but in autoimmunity it responds to the body's proteins itself, and the self antigens escape from the normal immune response of the body due to the phenomenon of immunological tolerance. So now there's another term called tolerance associated with the immune system. So I said the normal immune system, which should differentiate between the self and non-self cells or self and non-self tissues. Then the self-antigen will escape from the normal immune response of our body due to a phenomenon of immunological tolerance. Because we are having immunological tolerance for a normal person or a normal individual should have immunological tolerance. Means what? Our body should not fight against our cells. That is known as immunological response, immunological tolerance. And tolerance to the self-antigen is a normal feature of acquired immune system. And when this immunological tolerance is broken or failure of self-response happens, the body started acting against our own proteins and the cells and tissues in the body will be damaged. The host tissues or body parts will be affected, and it is actually a very rare phenomenon that happens, but it's very dangerous situation too. So autoimmunity results from the loss of self-tolerance. So autoimmunity causes because of loss of self-tolerance. You can't identify self and non-self proteins. And the different types are central tolerance and peripheral tolerance. So the immunity, the causes of this immunity, autoimmunity are maybe genetic. Autoimmunity results from the genetic disorders, or by birth itself you'll be having some defective genes that results in autoimmune disorder, or you'll be having altered immune cells. Second, it may be environmental. Some environmental radiations can cause autoimmunity. Some pathogens like viruses and bacteria can cause autoimmunity. What causes, basically what happens, the loss of self-tolerance is the cause of this autoimmunity. So self-antigens may normally be hidden from the immune system so as to prevent the autoimmune response. So these are the factors, genetic or environmental. So the autoimmunity classification can be, is of two types. It can be organ specific or it can be systemic. Or a particular system will be affected or a particular organ will be affected. Then the organ specific autoimmune diseases are diseases are the organ specific, means only one organ is affected. Systemic means the whole system will be affected. It can be a respiratory system, etc. like that. So organ specific autoimmune disorders are type 1 diabetes mellitus, then Goodpasture syndrome, multiple sclerosis, Graves' disease, Hashimoto thyroiditis, autoimmune pernicious anemia, autoimmune Addison's disease, vitiligo, then myasthenia gravis. Then systemic rheumatoid arthritis, scleroderma, and systemic lupus erythematosus. We'll see some examples of this organ-specific autoimmune diseases as well as systemic autoimmune disease. First is the organ, that is the autoimmune endocrine diseases, that is Hashimoto disease. The antibodies will fight against the thyroid peroxidase. Then primary myxedema causes the atrophy. Atrophy means it becomes smaller. Thyroid is affected. It is organ specific. Thyroid gland will become smaller in size. Then Graves' disease, the autoantibodies will kill the TSH. Then pancreas, it's organ specific, type 1 diabetes. Adrenal gland, it's affected by Addison's disease. Gonads, it is affected by autoimmune euphoritis, autoimmune orchitis. The pituitary gland is affected by lymphocytic hypophysitis. This is Graves' disease. One example of this, one example of the organ-specific autoimmune diseases. I said the organ-specific autoimmune disease means a specific organ is affected. It can be thyroid gland, it can be endocrine gland, it can be heart, it can be lungs, any specific. So organ specific or autoimmune disease involve a response directed against a single organ. A single organ will be affected by organ specific autoimmune disease. And this includes the disease in which the autoantibodies will bind to the self-antigen and resulting in the cellular damage. One example is Graves' disease. Example is Graves' disease, and in case of Graves' disease is also called exophthalmic goiter. There is another goiter which is caused due to the deficiency of what? Iodine. Deficiency of iodine caused goiter, and this Graves' disease is also called exophthalmic goiter. Exophthalmic. It's also called exophthalmic goiter. Exophthalmic goiter. Exophthalmic goiter or Graves' disease. And this autoimmune antibodies behave like the TSH. See what happens? The autoimmune antibodies or the antibodies in our body will behave like TSH. That's what is known as molecular mimicry. Molecular mimicry means it will mimic another molecule. That is called molecular mimicry. Molecular mimicry. Molecular mimicry. Our antibody will mimic a hormone in the body. That is called molecular mimicry. Molecular mimicry means mimicking a molecule. So in exophthalmic goiter, what happens? The autoantibodies. So our body's antibodies will behave like TSH or thyroid stimulating hormone. And as a result of this, what happens? Thyroid stimulating hormone. So what is the function of thyroid stimulating hormone? To stimulate the thyroid gland. So the thyroid gland will be stimulated continuously. See, because it is more, what happens? The thyroid gland will be stimulated uncontrollably or continuously, and this result in the enlargement of the thyroid gland. Thyroid gland will enlarge, and that will result in the bulging of eyes. This is a symptom or manifestation as a one of the prominent symptoms of the Graves' disease. So what is Graves' disease? It is also called exophthalmic goiter. It is an autoimmune disorder. What is it doing? The antibodies will mimic TSH or thyroid stimulating hormone. See, it will mimic TSH or thyroid stimulating hormone. So the target organ is thyroid. The target organ is thyroid. So this thyroid will be continuously activated by the antibody. Antibody mimic a TSH, and this antibody will continuously activate the thyroid, and the thyroid gland will become large, and as a result it will result in the bulging of eyes. Because the thyroid gland is continuously activated, what happens? The metabolic rate will be very high, the body temperature will rise, weight loss will be there. These people, because of the increased metabolic rate, these people will be very thin. Some people, some people, if they eat a lot, they will not gain weight, and that may be because of increased metabolic rate. And increased metabolic rate is because of the continuous activation of the thyroid gland. It doesn't mean that, this is actually a hypothesis, doesn't mean that all thin people are having Graves' disease or anything like that. They should have a bulged eyes. So weight loss, nervousness, then increased pulse. They have rapid pulse rates or increased heart rate. Then these are the symptoms of this exophthalmic goiter, and the treatment are anti-thyroid drugs like methimazole or tapazole is given, or radioactive iodine is given. Surgery is there. The next disorder is Hashimoto thyroiditis. You need to learn any two. Hashimoto's thyroiditis. thyroiditis is a tissue-specific autoimmune disease, and it also affects the thyroid gland. Hashimoto disease or Hashimoto thyroiditis also affects the thyroid gland. Graves' disease also affects the thyroid gland. Hashimoto's disease also affects the thyroid gland. And what happens? It affects the thyroid gland, in which the antibodies are produced against the thyroglobulin. In our bodies, antibodies will work against the thyroglobulin, and thyroglobulin are actually precursor molecules of thyroid hormones. Thyroxine, thyroglobulin is a protein which is a precursor of this thyroid hormone. So in Hashimoto's thyroiditis, what happens? The antibodies will fight with the thyroglobulin. Antibodies will fight with thyroglobulin. Thyroglobulin. And thyroglobulin will be affected here. Antibody will destroy thyroglobulin. So if there is no thyroglobulin, what happens? The hormones from thyroid gland will not be produced. Then, as a result of the metabolic rate will be affected. That is chronic lymphocytic thyroiditis, and the females are more susceptible to this Hashimoto's disease. 12:1. Then the effector mechanisms are thyroglobulin is affected, and this is the primary target. Thyroglobulin is affected, then thyroid peroxidase is affected, and this people will be having defective metabolic rate. They will not have a normal metabolic rate because our metabolism is controlled by the thyroid gland. So defective metabolism will be there. So these are the two organ-specific autoimmune disorder. Autoimmune disorder means what? Our body fight against our own cells. Organ-specific autoimmunity means what? A particular organ will be affected. First example was Graves' disease. The affected organ was thyroid gland. Next is Hashimoto's disease. Again, that is also a thyroid gland is affected in a different way. So, it is the symptoms are fatigue, cold intolerance. They cannot withstand cold, weight gain. They will be having a fatty nature, depression. They have enlarged thyroid gland. The T3, T4 will be decreased in their blood. TSH will be more. The thyroglobulin will be affected, and the treatment is actually the replacement therapy. The next is non-organ specific. Second type is non-organ specific or systemic. I said a system will be affected. That is called non-organ specific. Non-organ specific diseases are directed against a broad spectrum of tissues. Means not only a particular tissue is affected, but a number of tissues or a number of organs will be affected. A system will be affected. Example is the typical rheumatoid or joint deformities or rheumatoid arthritis, and the examples of non-organ specific disorders autoimmunity are multiple sclerosis, rheumatoid arthritis, and scleroderma. And this is a symptom. You can see here the picture shows the rheumatoid arthritis. And rheumatoid arthritis means, in Malayalam you say it's associated with called Vada. Rheumatoid arthritis means typically the joint deformities. You can see there is at the joints, at particular region, the body, you can see here a nodule is there in the small finger, this portion, and there is a deformity. Some region will be having a curve like this. There's no uniformity between the joints, and that is a manifestation of this rheumatoid arthritis. There is a wasting of the muscles. At some region the muscle will be thin, at some region it will be swollen, and they'll be having some nodules at some joints, and there will be a fingernail clubbing. It's or pulmonary fibrosis are some of the clinic or the observable symptoms in this rheumatoid arthritis. So these are organ specific. This is all about autoimmunity, organ specific, non-organ specific. Then we also learned about the immunodeficiency diseases, which are primary immunodeficiency diseases and secondary immunodeficiency disease, combined immunodeficiency diseases, and we learned about acquired immunodeficiency diseases, under which comes the HIV. And with this, I conclude. Thank you.